Oh, and I'll put up my one slide that I was going to share. Okay, how's that?
All right, so are you guys all ready? We can open up the waiting room.
Yeah.
All right. Yeah, it should be open now.
Okay. Hello, everyone. Welcome to the Prebiotic Chemistry and Early Earth Environment seminar series. Thank you for joining us today. My name is Danielle Simkis, and I am one of the seminar organizers, along with James Aguchi, Becca Guthmetzler, and Albert Fehrenbach. For those of you who have not attended a PCE3 seminar series before, this seminar series is held on Thursdays every 3 weeks, and the goal of the series is to facilitate science discussion across different research groups within the fields of prebiotic chemistry and early earth research. This year, for each of our seminars, we've selected an overarching theme for discussion. And so for today's session, the theme and the topic of focus will be protocells. The first portion of the protocells session today will consist of 3 presentations by our 3 guest speakers, Dr. Irene Chen from the University of California in Los Angeles, Dr. Claudia Bonfio from the Institute of Supramolecular Science and Engineering in France, and Dr. O'Flaherty from the University of Guelph in Canada. At the end of the session, after the 3 talks are finished, that's when we will open up the chat, and we'll use the chat as well as your microphones to ask questions, and we'll have a kind of a group Q&A discussion. And before we get started, we'd first like to invite you to participate in a very quick demographics poll that we send out at the start of each seminar session. Your participation, of course, is completely voluntary, but we just like to get a sense of who our audience is, what your field of study is, and where you're watching from. So hopefully on your screen you can see a poll, and we'll just take 20 seconds or so to fill this out. Okay, so I think we are ready to get started. I'll first stop sharing my screen. And Irene, if you'd like to get your slides up, just as a quick introduction, Dr. Irene Chen, she'll be presenting a brief topical overview of today's session theme of protocells. Irene Chen is an associate professor at UCLA. Oh, sorry, Irene, I think my voice is echoing through your... Yeah, there you go. Irene is an associate professor at UCLA in the Department of Chemical and Biomolecular Engineering and the Department of Chemistry and Biochemistry. She received a BA in chemistry and an MD PhD in biophysics from Harvard. Her research group studies lifelike biochemical systems to understand their fundamental properties and to address emerging challenges in biotechnology with a major focus on the biophysics and evolution of protocells. So Irene, whenever you're ready.
Okay, thanks Danielle. Thanks for the invitation and for the introduction. So I'm going to give a 5-minute introduction to protocells. That poll was really useful in showing that there's a pretty broad diversity of backgrounds and a good number of students here. So I'll just give a very broad overview of the field of protocells and what that means. So a protocell is a compartment that holds macromolecules for a living or proto-living system. And here's an illustration that some of you may have seen, which is created by Janet Owasa and Jack Shostak's group, of a possible model protocell encapsulated. We have this lipid bilayer encapsulating some RNA molecules. And that's one example of an existing model for protocells. So why do we need protocells? To me, there are two major reasons why protocells are important very early on in developing a living system. So one is the fact that the protocell presents a semi-permeable membrane. So some small molecules, nutrient molecules, waste products can go in and out of the protocell, while macromolecules like RNAs or proteins or other larger, less permeable solutes will be retained. So that's one feature of the protocell. But the second feature of protocells, which is also very important, is that it promotes the evolution of cooperative systems. So if you consider, let's say, a self-replicating RNA or some other similar system, a genetic system, you might ask, why does that need to be encapsulated in order to push it further along in evolution? And the reason is that if you have these RNAs or other molecules in free solution, so they're able to mix freely, then as soon as you get a phenotype from one of those molecules which is cooperative, for example, the ability to make copies of other molecules, that's a cooperative phenotype because that molecule is serving the function of copying other molecules. It increases the fitness of other molecules without directly increasing its own fitness. So if you have a feature, a property, an activity like that arising, then these molecules, these replicase molecules, will be copying these other molecules in the solution, so creating many copies of other molecules, but not directly helping itself. So this system in free solution will eventually run toward being overtaken by molecules that are really good at being copied, but are not very good at doing the copying. So that is Darwinian evolution, but it's not leading into what we would consider like a more complex, more interesting direction. So what do compartments do? What do protocells do? They enable the development of a second level of selection on top of the molecular level. So if we have compartmentalization of molecules, then we'll have some compartments that develop this new activity, and progeny compartments will contain more of this new activity. And compartments that, let's say, randomly segregate, but do not contain the activity will get lost because they don't have the ability to continue to replicate. And that's a form of so-called group selection where an altruistic or cooperative phenotype can be selected, even though within the compartment it doesn't do as well as the other molecules in there. As a compartment, because it increases the fitness of the entire compartment, those compartments do better than the other compartments that lack those cooperative phenotypes. So what types of systems are being used for modeling these protocells? So bilayer membranes, such as fatty acid vesicles, encapsulating RNAs or other molecules. These are a popular model of. People have also thought about using emulsion draw, usually a aqueous compartment, water in oil. Here's an illustration of that. Also serves the purpose of compartmentalization. And then maybe more recently, people are looking at liquid-liquid phase separation. It's a very old idea. Coacervates are an example of this LLPS phenomenon. But people have been increasingly interested in using these coacervates and other LLPS compartments as protocells. So some of the properties that people will consider when they are thinking about protocells and developing a model system for a protocell would be, are the components that I need prebiotically plausible, both in terms of the chemical synthesis and in terms of the conditions that are required for self-assembly. Can I make a self-reproducing system? So can we have some inheritance within of those compartments, and will they be able to grow and divide and continue to replicate as a compartment? What is the permeability of those compartments? Do they actually retain genetic material and let important monomers through? Do they have any interaction and interdependence with what is encapsulated? Or are they sort of an independently replicating system? At some point, we need to have the compartment communicating with the internal contents with the genetic material. So what are the mechanisms for that interaction? And at some point, then we'll also want those protocells to transition into real cells. And what does that transition look like? Could we create a plausible pathway from protocells to cells? And then there are probably there's certainly other considerations that people are thinking about that I haven't listed here, but those are some of the main ones. And if you would like a little bit more of a primer on this, if you've never come across protocells or thought about them before, then we recently wrote it's called a primer for current biology about protocells, to just give you an introduction and point towards some key references. And I'll leave you with this image. And if you want to learn more about what this image represents, please take a look at the paper. Thank you.
Great, thank you so much, Irene. That was a great introduction. We will move directly on to our next speaker, which is Dr. Claudia Bonfio. So as Claudia is getting her presentation up, I'll just do a quick introduction. So Claudia is a junior group leader at the Institute of Supramolecular Science and Engineering in Strasbourg, France. She obtained her PhD in biomolecular sciences at the University of Trento in Italy, during which she focused on the synthesis and activity of primitive catalysts on early Earth and spent research periods abroad as a visiting science student at Harvard. After her PhD, she worked in the MRC Laboratory of Molecular Biology in Cambridge and then the Department of Chemistry at the University of Cambridge, where she tackled fundamental questions related to the emergence of functional primitive cells. Currently, her group focuses on prebiotic supramolecular chemistry with a particular focus on interactions between supramolecular structures and biomolecules.
So thank you very much for the introduction, and thank you very much for the invitation. It's a pleasure to be here. Hi, I'm Claudia. It would be nice to see you all, but we'll make this work. So I would say that probably the first slides of my presentation are going to sort of mention things that most of you already know. But basically, what we know is that a lot of people are interested in understanding how life started on our planet, and we know that this is such an interdisciplinary field, and there's a lot of people that can tackle this question from a lot of different angles and with a lot of different approaches. So what is the approach that we are interested in, or that we are working on and applying in the lab, is basically focused on trying to understand how chemistry became biology. So how something that we definitely would not consider alive, molecules, simple molecules, more complex molecules, chemical reactions, how they can interact and cooperate to form more complex chemical systems that at some point end up forming something that we definitely would consider alive, for example, a living system or a living cell. So when we think about living cells, obviously we know, as we said, we know we are living, they are living. So we know that they have some properties, some characteristics that make them alive. And so what we can try to do is get inspiration from living cells and try to build something that looks like a living cell, something that has similar properties, something that has similar functionalities. And so Something that I thought was useful here was to give a definition of a term that I'm going to use a lot during my presentation, that Irene has already used and probably Derek will use as well. That is the main topic of today's webinar, which is protocell. So there's a lot of people that use this term, and so I figured that I could take this definition that Caparuit Mirazzo wrote down more than 10 years ago, because that explains what a protocell is. And so if you have a look at what he wrote, basically a protocell is any kind of model, and it could be both theoretical or experimental. And it's any kind of model that that involves two components: a compartment and chemical reactions that take place inside or around this compartment. And this combination between the chemical processes and the compartment gives something that that we hope can explain how modern cells might have emerged on early Earth. So the problem is that we have a definition of what a primitive cell or protocell is or could look like. This is what we would imagine if we had to draw down a primitive cell. We imagine a compartment. I quite like lipids, so I'm imagining a lipid-based compartment that where lipids would self-assemble into a membrane. I'm imagining chemical reactions that can take place inside of that. We tend to think of this as a very simple system, so very simple lipids that self-assemble into a lipid membrane, and very simple chemical reactions that take place inside this primitive cell. The problem is that when we actually go to look at modern cells, the situation is slightly different. Not only the chemical reactions that take place inside inside modern cells are not only many more compared to the number of chemical reactions we tend to consider for primitive cells, or we have explored so far for primitive cells, but also they are all interconnected with one another and with the lipid membrane. And even if we just look at the lipid membrane, so the compartment of modern cells, we can see that the complexity of the lipid membrane in modern cells is way, way, way higher compared to what we think about when we work with primitive cells. Not only in terms of lipid mixtures and the presence of different types of lipids, but also considering the fact that modern membranes are composed about of 50% lipids and 50% proteins. So when we want to tackle this challenge and we want to build a model protocell, not only the first point we need to address is, okay, how do we get all of the building blocks that we need to build a primitive cell, but also how do we make a primitive cell look like a modern cell? How do we go after we've made a primitive cell? How do we go about making something that looks more like the modern cells we have right now? So what we're going to focus on right now is mostly the first part. So how do we go about building a primitive cell? And the first question that I figure we could answer together is how early lipids, so the kind of lipids molecules that can self-assemble into a lipid membrane that were most likely available on early Earth, become the lipids that we have right now in our modern membranes. So we can imagine, to answer this question in a very simple way, we can imagine this as a treasure hunt map. And so if we have our treasure, this is a very simple schematic representation of a phospholipid, which is one of the major components of bacterial membranes right now. We can see that this molecule is composed of different parts. So we have usually an aliphatic chain or a hydrophobic part, which is usually made of fatty acids. We have a glycerol part that here is depicted in light blue. Then we have a phosphate here in orange, and then usually we have something else attached to the phosphate group, most commonly ammonium group. So here we have ethanol amine, but you can have serine, you can have choline, you can have a range of different molecules there. But this is more or less how a modern lipid looks like. Now, when we go and look at the kind of molecules we had on early Earth, we most likely had all the main components of this modern phospholipids. We need to find a way to combine all of them together. So on the left, you can see the starting point, which is fatty acids. Irene already mentioned them. So how do we go about combining all of this together? I tried to answer this question at the beginning of my postdoc when I was in Cambridge working with John Sutherland. And what we were curious about was to start from a molecule that can be considered prebiotically plausible, which is glycerol phosphate that you can see here in orange. And we decided to see what would happen if we were trying to add a fatty acid chain to this glycerol phosphate. So we activated fatty acids and other carboxylic acids, and we observed whether we would form mono and bis acylated species. So mono would be these blue species here, and bis acylated species would be these green species here. And what you can see on the right are NMR spectra that basically tell us how much this reaction is working. And what you can see is that regardless of the chain length of the acyl carrier, we could observe the formation of our products, so mono and bis acylated species. Obviously, what we were more interested in was something that would then self-assemble into lipid membranes. So when you have a sufficiently long chain on your acyl carrier, then you can form amphiphiles. Both mono and bis acylated species with sufficiently long chain behave as amphiphiles. They self-assemble into lipid membranes, as you can see in the picture. The problem with this is that if we go back to our NMR data, you can see that on the top, we have short chains, and the reaction is almost complete after 6 hours. But the more we go from top to bottom, the more we are increasing the chain length of the acyl carrier. And you can see that the reaction takes longer and longer and becomes less and less efficient. So this is a problem because you can imagine that on early Earth, you don't have a purified system as you would have in the lab, but you would have probably a mixture of different molecules. And so if we imagine that on early Earth, we had a mixture of fatty acids and activated fatty acids, then we would end up most likely with all the glycerol phosphate blocked into molecules that, like the ones on the top of the spectra, that would not be able to self-assemble. So we would lose our ability to form lipid membranes. So we needed to find a solution to this, and the solution that we found was based on the physicochemical properties of the molecules that we were forming. We decided to observe how stable they were in water, how fastly they would hydrolyze, so how easy the ester would get hydrolyzed and would yield back glycerol phosphate. And you can see here that the fastest molecule to hydrolyze is the short chain acetyl, the acetylated glycerol phosphate, while the longer chains would not hydrolyze very efficiently. And so this gave us an idea, because if we start from glycerol phosphate and we expose this glycerol phosphate to a range of activated carboxylic acid, some with sufficiently long chains, some with shorter chains, then yes, we would more efficiently form the short chain acylated products, but those would be also the ones that hydrolyze more efficiently, giving us back our substrate. While the ones with longer chains would not hydrolyze that efficiently, and they would potentially self-assemble into lipid membranes. And this is exactly what we observe. What you can see from left to right is an increasing number of cycles of this activation, acylation, and hydrolysis cycle that we designed. And you can see that we accumulated more and more of the self-assembling species, yielding to more and more lipid membranes formed in solution. So now if we go back to our treasure hunt map, we are sort of halfway through. We combined together 3 of the building blocks that are needed to make modern phospholipids, modern lipids. And something that we are trying to work on in the lab right now is how we combine the last building block in order to make lipids that look exactly like modern lipids. Now, it's not just about making the lipids, but it's also about understanding how these lipids, after they self-assemble, behave in solution. Because obviously we are not just making lipid membranes, but these lipid membranes can be quite sensitive to the pH we have in solution, the ionic strength we have in solution, and if we are changing these parameters, the lipid membranes might change as well. So I was particularly interested during the pandemic, actually. So this is a project that started 1 month before the pandemic. I was quite interested in understanding how lipid membranes would respond to environmental fluctuations and environmental changes. And something that got my attention was a paper that my PhD supervisor, Sherif Mensi, worked on, published when he was in Jack Shostak's group. And this is a paper that describes the stability of fatty acid-based membranes to different temperatures. So what you're seeing here is the ability of fatty acid membranes to keep their content when they are exposed at high temperature. And you can see that, for example, for decanoic acid-based vesicles, when you reach a temperature that is around 50 degrees, the content, in this case, they were looking at oligonucleotides. The oligonucleotides that were originally inside the lipid membrane get released and leak out of the solution up to a point in which at 70 degrees there's no more oligonucleotide inside the lipid membrane. So this is dependent on the lipid that is used. So for example, you can see here oleic acid based vesicles where the chain is longer and there's an additional double bond, and the chain are much more stable, so the leakage occurs at much higher temperature. It's also size dependent in terms of the content size, and you can see that smaller molecules leak out more efficiently compared to longer ones. So they focus very much on the content and what happens to the content of the primitive cell once it's heated. What I was curious about was what happens to the membrane per se. And so what I decided to do was basically repeating their experiment without caring at all about the content, but just looking at the membrane. So we prepared lipid membranes made of myristoleic acid. This is how they look like under confocal microscopy, and we decided to heat them. So what you are going to see now is a movie of fluorescently labeled myristoleic acid vesicles, and we are increasing the temperature. And what you see is that at some point the temperature becomes quite high and the lipid membranes are no longer stable. And what you see is that they collapse. They collapse into very dense structures that turns out are oil droplets. And these oil droplets keep coalescing and fusing and growing and growing. And then at some point, we decide to go back to room temperature. And what you'll see in a moment is that these oil droplets start to become quite unstable. And you'll see that when they reach a much lower temperature, from the surface of the oil droplets, new lipid membrane starts to bud out. So these are just some pictures of the movie I've just shown you. You can see from left to right, we are increasing the temperature. And so here on the left, we have lipid membranes. Here on the right, we have oil droplets. When we go back, you can see that from the oil droplet surface at the interface with the aqueous solution, you can see that there are new membranes that are budding out from the surface of the oil droplet. Now, this is something that is quite cool to look at through microscopy, but we can also study this in a more quantifiable way. So what you are seeing here are data that we observe by fluorescence, that we obtain by fluorescence and absorbance. The gray line, the gray profile, is what you get if you look at the turbidity of the solution, of a solution containing lipid membranes when you increase the temperature. And you see that in this specific case, we had a very strong increase in the turbidity of the solution after a certain temperature. A similar behavior can be observed by fluorescence. Here we were using a dye that is very sensitive to lipid packing. And at some point, at the same temperature, we observed that the lipid packing changes quite dramatically. And this is, again, the same pictures that we saw before. We have 25 degrees lipid membranes, 95 degrees oil droplets. Why is this happening? Well, it turns out that there are quite a lot of buffers that are quite sensitive to temperature. Their pKa changes with temperature. And this is something that happens with the buffer that we were using as well. You can see that tris buffer here in purple. If you start from room temperature with a pH of 8.5, the pH of the solution goes to about 6 when you are at 95 degrees. Now, this is something very well known, and it's not true for any kind of buffer. So there are different buffers that have different sensitivity to pH. You can see that phosphate buffer is relatively stable at higher temperature. And so what we decided to look at was different types of buffers and how lipid membranes made of fatty acids behave at different in different buffers. You can see that this is the kind of profile that we observe with tris buffer, with more prebiotically plausible buffers, for example, glycine and glycylglycine. You can see that the temperature at which we have this collapsing of lipid membranes into oil droplets is quite different. And you can also see that with phosphate buffer that, as we saw before, does not really change its pH when the temperature increases. We do not observe any collapsing at all. So lipid membranes, so if you just want to have a take-home message from this part of the talk, is that not necessarily primitive membranes are destroyed by high temperatures. They can be thermally stable. It really depends on the molecules you have in solution around the lipid membranes, and especially the buffer that you are using. Now, going back to what my PhD supervisor looked at, the content leakage, we decided to look at that as well. So we took some lipid membranes, put inside some fluorescent content. When we heat the membrane, when we heat the solution, the whole content gets released. And then when we go back to room temperature, some of the content gets reuptaken and some of that doesn't. The same happens if we have completely empty membranes at the beginning and we have only some fluorophore outside the lipid membrane. At the end of the cycle, we can see some of the fluorescent cargo getting encapsulated in the formation of the new membranes. So what we thought of was, okay, we can re-encapsulate the content after it gets released during the cycle. What if we have multiple populations of different primitive cells? So what we decided to do was to take two populations that contain two different fluorophores. We mix them together. We exposed them to this thermal cycle. So we heated them and then we cooled them down. And what you can see is that the new population of primitive cells that we formed had a mixed content deriving from the two parent primitive cells. The same is true for the lipid composition. If we start from lipid membranes that have that are made of different lipids, and then we mix them and expose them to this thermal cycle, the new population of primitive cells, the daughter protocells, have now a mixed composition of lipids that they took from their parent cells. So I thought of showing you a last story that probably is going to be also helpful then to smoothly move towards next talk. So we all talk about primitive cells, and we said at the beginning that the way we envision primitive cells is a compartment and chemistry that happens either inside or around these lipid membranes. So how do we know if this chemistry that we want to take place, that we want to run inside these primitive cells, is affecting at all, is having any effect on the compartment per se? So something that was developed in my in the group when I was postdoc was what we call activation chemistry. So in John Sutherland's group, a couple of colleagues of mine discovered that you can use methyl isocyanide to activate simultaneously carboxylates and phosphate groups to yield oligonucleotides, oligopeptides, and mixed species in which you have aminoacylated or peptidylated nucleotides. The point is that, so this would be the nucleotide, this would be representing an amino acid, but the problem is that activating carboxylates does not only mean activating amino acids. It can also activate fatty acids that we use for our membranes. So what we decided to do was doing a very simple experiment in which we took nucleotides, encapsulated them inside fatty acid-based membranes, exposed them to activation conditions. What you can see is that in the absence of vesicles, the nucleotide gets completely converted into its cyclic form, which is the outcome of the activation of A3'P. What happens when you repeat the experiment in the presence of decanoic acid-based vesicles is that most of the A3'P of the nucleotide gets converted, but we also have a new peak. And this new peak, we found out, is due to an acylated version of the nucleotide, which in this specific case means the formation of a fatty acylated nucleotide. Now, if you look at this molecule has a hydrophobic and a hydrophilic component. So the first thing that we decided to look into was whether this molecule would incorporate into lipid membrane and potentially would drive the growth of lipid membranes. And it turns out that this is exactly what happens. So if you start from lipid membranes and you add this lipid nucleotide conjugate, you induce the growth of lipid membranes. Now, this is not only happening when you have nucleotides, but you can also make fatty acylated amino acids, and they behave exactly in the same way. But what's interesting is that when you, instead of using nucleotides, you use fatty acids, of course, then you have the side chain of fatty acids to play with. And so in this case, we use arginine. And the reason we used arginine was because arginine bears a positive charge. And so what we ended up with was a conjugate made of a lipid and a positively charged amino acid. And so you can imagine that if we have a lipid membrane, and then we add this fatty acylated, positively charged amino acid conjugate, this would localize some positive charges on the lipid membrane. And these positive charges can be used to mediate the interaction between the lipid membrane and negatively charged species, for example, oligonucleotide present in solution. And so this is what we observed. You can see from the microscopy images that in the absence of the positively charged conjugate, we don't have any localization of RNA to the membrane, while we do observe a localization of the RNA to the membrane when we use these molecules. So this is it. This is what we've been working on over the past few years. So this is, if we go back to what we said before, more or less we have an idea of what kind of molecules we might have had on early Earth, how they could have assembled, how they could have responded to environmental conditions, how they can host chemical reactions and potentially get modified to form more functional primitive cells. What we are currently working on in the lab, and hopefully in a few years I'll be able to share with you the updates from this, is how we move from primitive cells and we make them looking more like modern cells. Now I have 2 slides of acknowledgements, and I know I'm 1 minute longer than expected, so I'm sorry about that. I have 2 slides of acknowledgements. The first one is for the people that really helped me to get to where I am. So my PhD supervisor, Sherif Mensi, my postdoc supervisors, John Sutherland and Chris Hunter, Jack Shostak, who's hosted me both during my PhD and during my postdoc, and especially the people that collaborated with me on the lipid membrane to oil droplet transition, especially Roger, who's a very creative PhD student, now postdoc at Cambridge University. And the second slide of acknowledgements is for my current group, where in Strasbourg, where in particular Maya, my first PhD student, is currently working on the synthesis of modern phospholipids. And with this, I thank you for hopefully listening to me, and I guess I leave the floor for Derek.
Great, thank you so much, Claudia. That was a really nice talk. So we'll move directly to Derek's talk next. Derek O'Flaherty is from Montreal, Canada. He completed an honors Bachelor of Science in Chemistry at Concordia University in Montreal and a PhD in the Wilds Lab at Concordia University. He then joined Professor Jack Shostak's research group as a postdoctoral fellow at Massachusetts General Hospital at Harvard University. He developed strategies to improve the non-enzymatic replication of genetic polymers as it pertains to the origins of life and the assembly of a synthetic cell. And in 2020, he became an assistant professor at the University of Guelph in Canada in the Department of Chemistry, where he is focusing on developing nucleic acid biotechnology and assembling a synthetic cell. So go ahead.
Thank you. Yeah, thank you so much for the introduction. I'm really excited that I could share a little bit of the research, and I think this will funnel well into, or at least Claudia's presentation will funnel well into my section. I realize I should have gone first because this is a really tough act to follow, but nonetheless, I'll give it a shot. So I'll be talking about non-enzymatic genome replication inside these model protocells, and my talk is divided into two sections or two pathways. The first one is polymerization, and I'll be talking a little bit about chemistry. So for organic chemists, I think this will be great, but for a non-organic chemist, I hope you'll be able to follow. Using these 3' amino nucleotides. Let me just put my laser pointer here. So these 3' amino nucleotides. And the second pathway I'll be talking about is the ligation pathway, where we use short fragments terminated with these modifications to assemble much longer products. So in terms of introduction, I think this might be a little bit basic for this audience, but what I'd like to show on this timeline that was pulled from one of Jerry Joyce's papers is the area of research that we focus on. So it's really at this frontier of pre-RNA world to RNA world. And the way I like to think about it is that, everything here is driven by chemistry. And then here we finally funneled into an enzymatic world that's primarily relying on RNA enzymatic function. And this transition could, there's many hypotheses that could allow this transition to happen, but I like to think about it through an RNA-like pathway. We often get questions like, why RNA? And so, again, I think this is quite basic, but for this audience, but we know that entire viruses encode their genome with RNA, so we know it can govern some type of inheritance. It can catalyze really useful chemical reactions like phosphodiester bond formation or phosphodiester bond breakage. The ribosome is a ribozyme, which means that the RNA is at its core and it's actually doing the catalysis, making it probably the most sophisticated ribozyme that we know of. And if you look at modern biology, there are many different cofactors that have an RNA unit that's kind of dangling on it, which really speaks to RNA being one of those molecular fossils. So going back to modern biology, when we think about genome replication, we often think about these nucleoside triphosphates. using these highly evolved and highly sophisticated polymerases, we can replicate genomes with pinpoint accuracy, and we can do so very, very quickly. But going back to early Earth, we wouldn't have access to these sophisticated catalysts. And so these nucleoside triphosphates turn out to be quite stable. And so much so that the order on which they spontaneously condense with one another or extend is on the order of RNA degradation. So they're really not useful all that much on prebiotic Earth without an enzyme. Now, luckily, pioneering work from Leslie Orgel discovered these phosphoimidazolides that now replace this pyrophosphate leaving group with this more activated or higher energy leaving group called 2-methylimidazole. This is actually version 2.0. The first one was just imidazole. And these can spontaneously react with one another, and this better leaving group restores that activity. And so looking at the replication cycle, you'll have to take my word for it, but we assume that the first template would have been present on early Earth. These monomers or activated monomers can bind on and off. When they're bound on, that can trigger a polymerization event. This could lead to full-length products. And to trigger a second cycle, now we got to take these two strands, rip them apart, and initiate another reaction event. Now, despite more than 50 years on this particular problem, we still have a lot of difficulties ahead of us. The process by which we do it currently in the lab is not sequence general, and we have a particular hard time copying AU-rich sequences. And the current gold standard is set up to 7 nucleotides in the RNA system. And so I'll just go over that data in a bit, because this came out from Lee's work, who was a very talented postdoc in the Shostak lab. He's now a professor. He discovered this 2-amino-imidazole leaving group using a small library of leaving group that he tested. And using these model experiments known as primer extension experiments, where you have a template, which is a longer strand, a shorter strand called the primer. You throw in your activated mononucleotides, you extend to full length or to whatever length it can extend to. We analyze these reactions using polyacrylamide gel electrophoresis, and this is an example here. This is a time course, so each lane represents a time point in the reaction. And as you increase in time, you can see the disappearance of the short strand and formation of longer products up here in a stepwise fashion. And so this is a one-pot reaction with thrown in these new leaving group activated mononucleotides. We have to use specific tricks like these auxiliaries that are binding downstream from where your monomer binds to allow this reaction to happen. But nonetheless, what Lee had shown is that you can copy all 4 letters in one pot. And this was great. Really what this report showed is that now we can copy sequences that are otherwise impossible to copy or had been impossible to copy. And that +7 experiment is shown here. This was a big step forward, but it is still not sequence general, meaning that if you swap, for instance, this G with this A in the sequence, you now enter kind of another few stalled products that doesn't give you the full length product as shown here. So my first project in the Shostak lab was looking into, can we apply what we had learned in the past 6 or 7 years to the compartment side of the lab. And I wasn't the first one to work on this. These are just seminal experiments that are trying to couple primer extension with compartments, so protocells. In this case, they're vesicles. Sharif Mansi had shown that you can get very good polymerization using these amino nucleotides. So you can see this 2' amino group here. And Kate Adamella had shown that you can make an RNA copying process compatible with compartments if you chelate the magnesium. Because one thing that's a bit of an issue with these vesicles or lipid-based vesicles is that they're not stable to magnesium. Magnesium actually destroys them and precipitates them. So using this citrate chelated magnesium, she was able to show that you can get some copying inside these model protocells. But mainly, both of these examples were focusing on what we call homotemplates. So that's when this region here is kept constant, and you're just adding 1 or 2 different monomers, but not all 4. So the challenge was as follows: can we copy mixed sequences within our model protocells? And the biggest challenge was, how do we get these activated trimers that are necessary for sequence general copying inside our vesicles? So this had been a project that I continued from early work by Neha Kamat, who's now at Northwestern. She had looked at what does the magnesium citrate do to the membrane in terms of permeability, and she had shown that now these activated trimers were able to passively diffuse through the membrane, which would give us that one key feature for us to allow this mixed template copying. But before I dive into the results, I just want to go over the experimental procedure, because these experiments are definitely not trivial. You first take your primer template duplex, you encapsulate it in vesicles. You do 1 quick round of purification and collect just your vesicle fraction. You then throw in your activated nucleotides. These can then participate in primary extension experiments. You have to re-purify at each given time point, clean up the sample to remove as much of these lipids as you can, and only then can you run that electrophoresis experiment. So something that normally takes you about 1 day to do when you're not doing it in vesicles, now can take you about 3 to 4 to even 7 days. So yeah, much more laborious. And here I'm showing 3 different types of membranes that we looked at. Probably the only one that's really prebiotically plausible is the C10 system. C14 and C18 were used as comparison. What I really want to gather from this is that we're increasing in length, which means that the thickness of the bilayer increases from left to right. Okay, so these, I'm just hopping into the results here. These are our experimental setup. We have our vesicles, our primer template are in. We want these activated trimers and monomers to come in and extend to full length. And so there's a lot going on this slide. I'm just going to walk you through it. Here we have on the top 2 panels just the addition of monomers. And what you can see is that the primer only gets to about +2, not very well at that, after 48 hours. And this is in solution. When you put this in compartments, it's even worse. But now if you have the trimer that's we know is required to get full length extension, here you can see full length extension. And because we had the magnesium citrate that enhances permeability, now we can get full length extension of all 4 nucleotides inside our model protocells. And this is just a schematic graphical representation of all the data. We included tricks like having a heat pulse at the beginning with our primary extension mixture. This seemed to aid, especially the kinetics at earlier time points, but not so much the levels at the later time points. So this was great. We got now up to 5 nucleotides in length. We could never reach that 7 nucleotide in compartments. I know I tried very hard, but I couldn't reach that same benchmark. That still gives us a problem, because we want to be around the 30 nucleotide length for extension if we want to see something interesting happening with our RNA duplex. Something that's 30 nucleotides in length is probably the target that we want to try and aim for. So as a bioorganic chemist, I thought, well, we can improve the chemical copying in one of 3 ways. We can play around with the leaving group, but Lee had already done that, and so I didn't really want to focus on that. We can play around with the nucleophile of the reaction, so these 2 hydroxyls, or you can play around with the scaffolding. So here you would put some kind of chemical modification on your base to enhance the binding, and hopefully that would help your chemical copying process. But in essence, we're thinking about these alternate genetic polymers. And the one that I focused a lot of my postdoc and what I'm currently working on these days is this NP system. So the NP system is a 3' aminonucleotide where you've swapped out the hydroxyl of DNA with an amino group in this sugar scaffold. So really, the NP system is a chemical mimic to DNA because we're just switching the nucleophile. But in reality, it's a structural mimic to RNA. And the reason being is that in solution, this adopts a specific sugar pucker that resembles much more RNA than DNA. And actually, we do have some crystal structures of polymers that are composed of these NP systems, and they resemble much more RNA than they do DNA. So I'll spare you the details on most of the organic chemistry that went into making these activated mononucleotides. Again, I wasn't the first to look at these aminonucleotides. There had been 2 reports from the Shostak group at the time when I joined that were very pertinent, that had looked at the 2-methyl-imidazole leaving group, but now I wanted to investigate the 2-amino-imidazole leaving group. I spent quite some time developing a general synthetic pathway to generate all these nucleotides in a scalable fashion that didn't require any fancy purification or very, very small scale reactions. And so once I had all 4 nucleotides in hand, we did a seminal experiment, I guess, a test trial, if you'd rather, with the sequence that Lee had copied quite well. And to our surprise, we got very good extension. In this case, with only the nucleotide present. We didn't have any of the trimer assistance or those fancy auxiliaries that was required in the RNA system. These are just the 4 nucleotides added together. And what you can see is within 1 day, full disappearance of the primer and full product formation of this +7. So we were very excited about this. This told us that this was an efficient process, but maybe not an accurate one. So we took our full-length product and tested it by LCMS and saw that the only detectable species that we could see was the primer plus the correct composition of nucleotides. Now, obviously, this doesn't give us the order of nucleotides, right? This just gives us that the product has 3 T's, 3 G's, 1 A, and 1 C. But we had other supporting data that showed that if you do have a mismatch, this severely stalls the reaction at that given mismatch step. So we thought it was a pretty accurate process. Now, moving into membrane compartments, this is the setup of the experiment. Note that we don't need those trimers. In this case, we recycled that same sequence, did the whole processing of your vesicle reaction. We have the 3 membranes shown here, increasing in thickness. And what you can see from this time course is that as you move from thicker to thicker, the kinetics slows down, which hints that really what's controlling the kinetics of this reaction is monomer permeability. And so this is summarized in this graph here. Nonetheless, after 3 days, we have very good extension of the full-length product. Okay, so at the same time that I was looking at the polymerization pathway, a very good colleague, Lijun Zhu, was looking at the ligation pathway of fragments that have an aminonucleotide at the terminal end. And so the ligation pathway is very attractive because now you only need a certain amount of reaction steps to get much, much longer sequences. You can presumably copy through secondary structures. So if this template region was big enough, then maybe it could do some type of self-pairing if it wraps around through, for instance, a loop motif. These secondary structures should be able to be denatured by competing fragments here. And for AU-rich sequences, this should not be an issue because binding shouldn't be the rate-limiting step in the process, simply because now you have much bigger fragments that are binding to your template. This pathway doesn't come without its drawbacks, so we expect that if it's an error-prone reaction, this would be further enhanced in the ligation pathway, because now, mutations that would be probably in the inner part of the ligator would still undergo reaction. So this could lead to what's called an inheritance catastrophe. It isn't prebiotically plausible to look at just ligation on its own without the presence of monomers. And it might not be compatible with compartments, because we had already established that these have to get through the membrane, but perhaps this is not as big of an issue as we thought it was. But when we initiated this project, we did a thorough literature search, and we found that just prebiotic investigation of ligation is pretty scarce. So we started by just looking at what the ligation pathway could offer, both in the RNA system, that's the top here, versus one that had the 3' amino. And what we were able to show is that the kinetics increased by more than 150-fold for running these Michaelis-Menten kinetics. And so we were happy with the fact that we could get the 3' amino to move forward. Next question that we asked was whether we can copy something that's +16 with 4 different tetramers, and sure enough, this worked very well. We did it without the presence of magnesium and in the presence of magnesium. And what you can see from the 2 panels is that magnesium really helps the catalyze that last step. And this, as you can see, is an A-rich ligator, which means that the binding would probably be quite weak. We do have supporting information that supports if you have a longer ligator that's composed of A's and U's, which bind presumably weaker, that this issue is not felt as much. And so yes, here we showed that what's limiting that last step is the binding of this ligator. But how do we make a ligation pathway compatible with compartmentalization was an experiment that we tried to engineer for quite some time. And so we knew that anything larger than tetramers would have a hard time getting into the membrane, getting through the membrane, I should say. So we were limited at tetramers. And if we use tetramers, then we could have a splint ligation pathway where the ligators are not, the splints, are not activated. So these would be only weakly bound. The top strand shown in blue are activated. So these tetramers would be reactive to one another. And these could assemble in almost like a Lego fashion where they just kind of click with one another. But the template, because they're not activated, these don't react and can easily fall off. And so what ends up happening is after these ligators enter the protocell, we have here this primer template duplex is used just as a means of monitoring the reaction. So it's more of an experimental limitation. If you take this away, the experiment presumably still works, but we needed a way to track the experiment. So this primer template duplex is already encapsulated. These tetramers, some activated, some not activated, would passively diffuse inside the membrane. They would assemble as shown up top here, and you'd get only the blue strand forming. And so having such an experiment could provide a solution to something called the strand displacement problem. And also, alluding to that introductory slide that I talked about, where I stated you'll have to take my word for it, this could give you a pathway for the first long template. And so we first set this up in solution, and we were very pleased to see that, within a few days, or actually this is just one day, we were observing products that were as long as 200 nucleotides in length, and this was great. I haven't really talked about these additives. Magnesium I had a little bit, but this intercalator ethinium bromide, I haven't really talked about that, and I'm happy to address any questions concerning this in a bit. But we were very excited to see, the production of these longer products. We also had stepwise additions of all these other products, which gives you a library length of different products, which is also quite interesting in a prebiotic setting. But what was more surprising is that we can make this process compatible with vesicles. And it might be a little bit difficult to see on your screen, but you do get the formation of these lengthier products as well. Okay, so with that, I'd just like to wrap up. I hope I convince you that the NP system has vast potential in the prebiotic realm. It's both efficient at the polymerization state and the ligation state, and that these processes are compatible with vesicular systems. So currently in my group, we're trying to improve the synthesis of these NP-DNA substrates by solid phase synthesis. These are quite challenging to synthesize with current methods. So we're trying to engineer new ways to synthesize these polymers so that we can study them to a greater extent. And also we're trying to show some kind of transition for a means to couple chemical copying to protocellular function. And so what I mean here is that it's great that we can get these monomers to participate in non-enzymatic primer extension, but once you get a full-length product, how do we get to something that gives off some type of structure or function phenotype to that group of protocells? And so I can think of this in one of three ways. We could have something like a chemical output, maybe formation of small molecules that aid, either has a feedback aid for replication, or maybe they localize to the membrane to give it additional sturdiness or something of the sorts. Photochemical features like lighting up to allow us to better track these populations, or we could even think about structural features. So generating some kind of products that could localize to the membrane and make those membranes more robust. Okay, with that, there are many people I'd like to acknowledge. Most of this work is pulled from when I was in the Shostak group. So obviously, Professor Shostak, awesome mentor, really enjoyed working in his group. My partner in crime, Lijun Zhu, who everything in this presentation is equal contributor, great colleague, and yeah, so kudos to her, lab colleagues, all these funding agencies that have supported these wonderful projects, the University of Guelph, my current group, who's absolutely fantastic. These funding agencies that are currently funding our Origins of Life initiatives and other initiatives. And with that, your attention as well. Thank you.
Awesome. Thank you so much, Derek.
Of course.
Three great talks. We're going to move directly into the Q&A session now. So the chat is open, so you're welcome to type in any questions that you have there. You're also welcome to raise your hand and ask your question live. So right away, we have a question from Kevin Devine. So I will ask you to unmute. You can ask your question.
Okay, thank you. Thank you, Claudia and Derek, for the two great talks. I mean, Derek, I just want to pull you up about your Origins of Life timeline. I think it might need a little updating in the light of very recent evidence.
Okay.
I would say prebiotic chemistry to RNA world will be more likely 4.3 to 4 billion years. And okay. I would say that. And then life as we know it, DNA, RNA, and protein, more likely 3.9 billion years.
3.9. Okay, also, perfect.
And about your nucleoside triphosphates, have you seen the latest papers coming out from my also similarly awesome supervisor, Stephen Benner's lab, about the prebiotic synthesis of RNA triphosphates and subsequently the synthesis of polyRNA from nucleoside triphosphates on prebiotic rock glasses? Have you seen those papers?
Yeah. So here you're talking about the wet dry cycles and the better paper that came out, I think not even a year ago, right? Within right. I think it was 2022. Yeah. I think these wet dry cycles are great in terms of kickstarting.
It actually gives you viable amounts of triphosphates and polyRNAs to play with, which could then be taken up by protocells.
Yes. Now the main issue arises from sequence diversity, right? So there and again, I haven't looked at the data to closer extent, but I think you have large biases to like poly-A's and poly-U sequences that I don't know how would translate into something that would be more sequence general.
I mean, I think that's initially. I think now the sequences are more diverse. I have to say, the latest evidence coming out of there is more diverse. So, I mean, it gives us a sort of where you don't need to invoke, imidazole activated nucleotides. No, I have to say. So I think this could be a shortcut to available RNA building blocks, because we got those. That was a big problem. That was, remember Gerald Joyce famously describing RNA as a prebiotic chemist's nightmare.
Right.
Now we know that's not necessarily the case. So
right.
I would say so. But having said, and also your 2' amino, nucleotides. You're right. They are structurally very similar to RNA than DNA. But I mean, as far as them being, do you think they were a part of the prebiotic landscape as well in terms of nucleotide chemistry? What's your view on that? Or is this just a sort of is this just one of a man-made concept? No.
Yeah. So yes, there's no prebiotic report at the moment that supports them being, present on early Earth or anything like that. We were using them as a proxy in terms of, well, RNA is difficult to study because it's so slow. Can we at least get an insight? What happens if that 3' hydroxyl, if it was made more nucleophilic? Yeah,
you're right, and more basic as well.
That's right. So that was more the angle that we were taking. Now, there are some convincing reports that are coming out of different prebiotic groups. So there's one that recently came out from the Pounder group on the 3' amino TNA system, I think. So I don't think it's, I don't think it's also accurate to completely dismiss them because there's no prebiotic plausible.
No, I agree. No,
of course, right?
Right. Okay. Thank you, Derek. It was very interesting.
Okay, great. Thank you. We have a question from Helen. I'll ask you to unmute.
Okay. Thank you. So I posted a link to my favorite semi-enclosed compartment, which is, of course, the spaces between mica sheets. And in figure 8, I describe how that has the option to protect one from parasite molecules. I also have a question for all of you. All types of living systems have high intracellular potassium, and it's not in seawater, it's not in fresh water. It seems like it must have been present at the origin of life if all living systems have it. And you guys are in a great place because you can put potassium in your test tubes. So do any of you have comments about how your science works in high potassium like physiological concentrations?
So I can speak about the non-enzymatic primary extension. It doesn't influence much for monovalent cations. You'll get a nice boost in reactivity, but it doesn't affect any of the chemistry there. We normally keep I think 10 millimolar concentration of potassium.
So that would be tiny compared to 100 millimolar physiological.
Yeah, right. It could only enhance it.
Wow, enhancement. But at any rate, so your system has no problem with potassium. And I don't know whether the other speakers' systems have problems with potassium. Thank you, Derek.
Yeah, so I would say that, so I haven't worked specifically with potassium. What is reported in literature is that especially in the case of fatty acid membranes, high ionic strands are not good. And when it comes to sodium chloride, that has been studied much more than potassium chloride, when we are in the range of the 100 millimolar concentration, you can start to have problems with the stability of lipid membranes made of fatty acids. So to some extent, monovalent cations are better than divalent cations, but very high concentrations are not ideal. This is true for fatty acid membranes. When it comes to phospholipid membranes, there's probably less of an effect of high concentrations. I would say I haven't specifically worked on that, but something else that I found quite interesting was that when I was in Jack Shostak's group working with fatty acid membranes, we would tend to use potassium hydroxide to pH solutions rather than sodium hydroxides, because the permeability and the stability of the stability of the membranes seemed to be higher when we were using potassium instead of sodium. We are still talking about much lower concentrations, the physiological concentrations of potassium that you mentioned before, but still there might be some kind of effect also on the type of cation we're talking about. But I would say when it goes to 100 or 200 millimolar, the instability starts to play quite a big role for fatty acid membranes.
Thank you. Yeah, I'm shocked that sort of like sodium, people work with stuff that's intracellular and they automatically add sodium instead of potassium for some dumb reason, in my not humble opinion. Okay, Derek.
Yeah, just to add on, Claudia, what you were saying, I just forgot something. When we do the non-enzymatic primary extension inside vesicles in the presence of chelated magnesium, we use potassium citrate, which is at 200 millimolar. Now, I don't know what that does to the membrane, and these were MAGMM vesicles, so I don't know if that plays into the stability, but they were also, I used the same type of concentration for decanoic acid vesicles. So I don't know if that has any impact.
Well, relevance. I'm delighted to hear that some people are putting potassium into their test tubes. Seems like progress. Thank you.
Thank you. So Helen and Kevin, if you have any other questions, please feel free to ask. Otherwise, your hands are still.
Helen, do you want to go first?
No, I think it's just you.
Okay, yeah. Got a question for Claudia. I really liked the data you showed when you had the combination of nucleotide and 2' fatty acid, that conjugate, and the effect it had on membrane formation. The data you showed was with adenosine. Have you looked at the other 3 nucleobase conjugates as well? And indeed, following on from that, have you looked at mixed sort of like nucleotide conjugates and the effects that they have?
Yeah, so the short answer is that we haven't done it yet. We are in the process of doing that in collaboration with a former postdoc from John Sutherland's group. So hopefully in a few months we will have some results. What we have done instead is we've started looking into conjugates made with different lipid chains. And so we are testing how much when these molecules start to play, when they start to integrate inside the membrane, and when they start to destabilize or stabilize the membrane. We are very interested in looking at what you were mentioning, and hopefully we'll have some data soon.
Because you've also got the potential for Watson-Crick base pairing forming inside your vesicles as well, which is very interesting.
Yeah, I agree.
Yeah, thank you. Excellent. Excellent talk.
Thanks.
Thanks. And Irene, do you have a question or comment?
I guess a little of both, sorry. So first, thank you, Helen, for commenting on the surface association. That's definitely a way to get around the parasite problem. I didn't include it in my intro just because they're not typically considered protocells, but they are a possible solution to that evolutionary problem. And then my question was for Claudia. You showed some really cool pictures where through this thermal cycling, you segregate into two populations where you get some vesicles which are highly concentrated with the contents and then some vesicles which are like empty. And I wonder if you could explain what's happening there.
Yeah, so that's not exactly what's happening in the sense that, no, I mean, you are totally right. So the pictures, the images that I show seem to give the impression that the fluorescence of the solution is much lower compared to the fluorescence of what's inside the lipid membrane. This is just an artifact due to the fact that we had to dilute the sample when we were taking the images. And so we diluted the fluorescence of the fluorophore in the solution. We did not dilute the one inside. So that's why they looked much more bright. So yeah, it was just an artifact, really.
Sorry, but you still have some which look bright and some which don't.
Yeah. No, yeah, that's very true. So I try to understand how the re-encapsulation works. So what we see when we try to visualize the whole process is that these oil droplets seem to start forming some onion-like structures. So you have a central oil droplet core, and then you start to have a lot of different layers. Then the process is very fast, the reformation of the membranes. Unclear whether the membranes only form when they are at the interface between the onion, let's say, and the bulk, the solution, or whether they also form inside these, among the layer, between the layers. So if that's what happens, then possibly those that form among the layers will be empty, while the ones that are exposed to the fluorescent solution would encapsulate that. So that might be a possible explanation. Something else we are trying to understand is how we can improve the encapsulation efficiency, because we actually end up with many more empty vesicles compared to the filled vesicles. And so we are trying to see in collaboration with Derek also how we can anchor the content, for example, some fluorescent oligo, to the lipid by using these conjugates that we've been looking at before, to see if we can form these oil droplets decorated with the DNA or with the RNA, whatever, so that then we favor the re-encapsulation. But this is also something that we hopefully will do soon.
That's really cool. Thank you.
Thanks.
Great, thank you. We have a question from Anu Raj. I hope I'm not mispronouncing that. I'll ask you to unmute.
Hello, am I audible?
Yeah, we can hear you. It's a little bit quiet, but.
So hello from me. I'm still a student from Calcutta University Biochemistry Department. Actually, when I saw that there was discussion about 2-amino-imidazole side chain of portions, so I need to just ask one thing, that we had a paper, we had to read a paper. It was also given in the Garrett and Grasham book in the RNA and DNA, means nucleic acid chapter. And in that chapter, they were talking about that amino-imidazole carbonitrile, that was the first precursor compound of adenine. So was that the reason to take an amino-imidazole portion in that nucleotide? I mean,
so I'm not sure if I understood the question correctly. Are you talking about the 2-amino-imidazole and its implication as a prebiotic molecule? Maybe I could just give you a little bit of background. So Albert Fehrenbach and Konstantin Giurgiu developed the prebiotic chemistry for 2-aminoimidazole from the same synthetic pathway as aminooxazole. And so that's, I think, a 2017 JAX, and that's why it has prebiotic relevance, or at least it gives support as to why it has prebiotic relevance. I'm not sure if I'm answering your question.
And yeah, actually I was asking that there was mentioning about the precursor of adenine, the base adenine. It was amidoimidazole carbonitrile. So was that any reason that there was a mention of 2-aminoimidazole, because there was something precursor in that end as aminoimidazole carbonitrile? So can it be a reason that in case of prebiotic chemistry they have designed that system in that way because there was a precursor aminoimidazole carbonitrile of adenine?
I don't think the 2-aminoimidazole can be synthesized from that same precursor. So I think it starts from, I think, either glyceraldehyde or glycoaldehyde in the presence of large amounts of ammonium chloride. I can't remember the details, but I don't think they're the same pathway. They might use similar feedstocks, but they're not the same pathway.
Okay, thank you.
Okay. And so questions like that are a little bit more technical, they could always be a good type of question to follow up on in our Slack group, which we will we'll include a link to it in the chat. So you can join the PC3 Slack group if you haven't already. And then that's where we have some post-seminar discussion about any topics that didn't quite get thoroughly discussed. So, so yeah, we'll post that in a moment. And then meanwhile, does anyone else have any questions? Because if not, we are just a couple of minutes before 2:30, so it's a good time for us to stop as well. Yeah, so I think we will just conclude then. So thank you so much to our three speakers. Those were really great talks. Thank you, Claudia, for staying late a little bit. She's in France, so it was a bit of a later presentation for her. And thank you, everyone, for attending. It was a really good session and good discussion, and we're really looking forward to seeing you in 3 weeks. So thank you so much.
Thanks,
Thank you.
Danielle.
Thank you.